Okay, we're on.
Okay, great. Perfect. Okay, so we are recording now. We are going to look at analysis and plotting in NetPyNE. And yeah, there's a lot of different analyses you can do already built in. And then obviously, as you were working with Salvador earlier, you can access all the data in NetPyNE to create your custom analyses and plots and things. But for now, we're just going to go through the built-ins. So our first step is to install Neuron and NetPyNE.
Joe, one question.
Yeah.
If we have to do analysis using a model DB, some model DB, can't you show some example using that? Like how to pull that from model DB to like NetPyNE?
Sure, we can. Well, we can do that later. I kind of designed this around GUI tutorial 3 just because it's got a lot of interesting stuff going on we could play around with.
Okay.
But yeah, definitely. We could definitely do a session on downloading something from ModelDB and getting it into NetPyNE.
Right. Thanks.
Yep.
Well, I have a question also. I see that on the collab you have like !NRNIV model and then space mod. What exactly is that doing? I know it's a terminal command, but I'm just...
Yeah, exactly. Yeah, we're about to get there. So first we want to clone. We've got a repository, the NetPyNE workspace.
Yeah.
Where we keep the GUI tutorials and stuff. So we're going to clone that so we have access to those mod files. And then we're going to change into the directory that was just created, change into the NetPyNE workspace. And now in this NetPyNE workspace, is the GUIs and a directory of mod files. And in this mod file directory is just all the the mechanisms that aren't built into Neuron. And so these have to be compiled on whatever machine you're using in order to get the sort of fast simulations Neuron does. So yeah. So yeah, we change into that workspace, and then this NRNIV MODL compiles those mod files. There's a bunch of them for it to go through. And at the end, we successfully created x86-64 special. So yeah, so now we're going to, this is essentially GUI tutorial 3, except I made a couple modifications for this session. Yeah, well, let's just take a quick look at it here. So, it's got the size, 2 types of cells that are uploaded from a file, broken into 3 layers, layer 2, layer 3, layer 4, and layer 5, with just 10 cells in each, an excitatory and an inhibitory mechanism, got some background stimulation, and we've got E to all connectivity and I to E connectivity. We've got the RxD, the reaction diffusion stuff in this model, so that way we can play around with those analyses and figures. Yeah, the cytosol, calcium and IP3. So that's RxD. And in our sim config, it's going to go for 1,000 milliseconds. And yeah, so the changes I made are down here at the bottom. It's got a bunch of analyses that were sort of automated every time you run it. I just commented those out to get rid of them. Yeah, so there's today we're just going to do the static plots, the static figures. But for almost every static analysis figure we can make, there's also an interactive version, which makes those HTML active interactive plots like you see on the GUI. That's using the interactive plotting directly from NetPyNE. So you can create those cool HTML plots, but we'll get into that later. For now, we're going to leave the eyes off our plots and just do the static. And I changed it. Before we were just recording from a single cell. And in order to show the power of our plot traces, I wanted to record from a bunch of cells. And yeah, so in this plot traces, the include has to be a list, which means it's in square brackets. And then this is a cool way to get the first cell from every population, E2 cell 0, not the GID, not the global ID 0, just the first cell in that population. So we get the first cell from each population recorded, and that way we can explore those traces. I'm going to shift enter in that. And then, yeah, so then we will run the simulation. So we've got to import sim and then create, simulate, analyze. This will probably take about a minute. And yeah, while that's running, so yeah, we've got a package index in NetPyNE. I got the link there. And yeah, so this this documentation is built from the doc strings in our code, and so yeah, it's kind of cool. So in the code, every function has a doc string, which is just a Python, string in Python, but it's got a set format that explains what's going on in that function. And so, for example, let's look at spikes is the most common. Analyses involve spikes. You can see we've got the stubs in place for a lot of stuff, but the information is there, even if it's missing a few things. Let's look at, where's our raster? Plot raster is probably one of the most common plots we're going to do. And yeah, so it's got, every option that you can feed into it. And then it describes those and gives you the defaults and then what other options you can put in there. And good, yeah, this successfully completed. So yeah, so now we'll begin exploring some analyses. Just so I don't have to type sim.analysis every time, I'm going to say SA equals sim.analysis, and then I can just do, if you do SA plot and wait a second, should come up. Yeah, it should come up with all the available plots there. And crap, what was I going to?
Can you zoom in on Colab?
Yes.
Thank you.
Yeah, I think this was plot 2D net. So yeah, we're going to SA plot 2D net. And almost every analysis function in NetPyNE has defaults set up. So you don't have to put anything in for most functions. And you'll get, sort of the most common outputs or most common settings. So yeah, so we did plot 2D net. Spits up this nice figure. It gives a legend for your cell populations. And so on. But there is a lot of, we're going to go to analysis.network, look at plot 2D net to see what all we've got. First thing you'll notice is this include option, okay? The default is all cells. So every cell in the model is included by default. But we're going to plot 2D net. For me. And instead of all, let's put, we don't really have any net stims. We can do a single population, or you could do single cells. Let's just do, we'll just do the excitatory populations to see what's in there. And this is a list, not a string, excuse me. And that list will include E2, E4, and E5. Shift enter. I gotta remember that semicolon at the end of everything. Okay, so now we've just got the excitatory populations. So you can, manipulate whatever you want to see. You can leave out stuff. and so on. There's different views. So the default is XY, but we can also do XZ. So let's change out the include. And yeah, if you guys want to keep all these, you can just do a new tab every time, and you'll be able to keep all this stuff. I'm going to keep doing it in the same cell, just so it doesn't get too crazy in here. Well, no, maybe I should. Yeah, I'm gonna do it in a new cell each time. That way it'll stay, and we can save it at the end. So yeah, so we want to do sa.plot 2D net. Plot 2D net. And now we want the view to be, why was an XZ was the other option. Shift, I didn't do the semicolon. Fortunately, it's not too much data. Yeah, so now you can see this is Z and X. So instead of looking at it vertically, now it's sort of top down. So yeah, the show cons is an option. So you can, the connections are shown by lines connecting everything. The GUI, if there are too many lines, automatically shuts off show cons. But for static plots, it's usually fine to include them. The default is true. You can put in the colors you want to use for your populations. So if you wanted to do this, you would make a dictionary where the key is the name of a population and the value is a color, RGB or hex. And then yeah, the plot will use that color instead of just the defaults that come up. You can change the fig size, the font size, save the data. So yeah, so every time you make this plot, and you just see it spits out all those numbers. That's actually all the data you need to make this plot. So you can use this plotting function to get out the data and then use it wherever you want. You can save the figure, true or false. If you set it to true, it'll give it the name of your simulation and underscore 2D net or whatever. Or you can actually put in a string if you want to give it a specific name, save it to a specific name. And you can save it as PNG, JPEGs, EPS, or TIFF files. Show fig, the default is true because normally when you plot a figure, you want to show it. But a lot of times you just want to save a figure. You don't actually want to see it. So you can set show fig to false. There's a little problem in the doc string, so the formatting went a little wonky here. So there's a typo in my doc string somewhere that I'll have to fix. Yeah, so that's plot 2D net. Moving on, let's look at plot con. So creating the connectivity matrix. So yeah, so this is the default. If you don't put in anything, you get all your populations and your stims. And again, pre-synaptic on this side, post-synaptic on this side. And the strength is like the average weight. So a weight is between any two cells. Between any two populations, you average that weight and you call it strength. We're going to go to, back to network and look at plot con. And here, instead of include, we've got include pre and include post. Because you can have all of them on the pre and just one on the post if that's what you wanted to see. And we'll play around with that. Let's see, feature. Yeah, the feature is important. We want to talk about the order by, okay. Yeah, so first let's just play around with include pre and include post. So in here, I'm going to say include pre equals, it's going to be a list, and include post equals, and that's going to be a list. And on the pre, I'm going to put the excitatory E2, E4, E5. And on the post, we'll just put the inhibitory I2, capital I4, I5, and semicolon at the end. Shift Enter. Great. So now we see on the pre, we've just got our excitatory populations. On the post, we've just got our inhibitory. The strength isn't the only thing we want to look at. You can also look at the weight, which if you're just looking at between two cells, you have to do the weight because that's all there is between individual cells. You can look at the delay, you can look at the number of connections, and So let's look at the number of connections. It's kind of a fun one. So we're going to do plot raster, but this time we're going to do feature equals num cons. What is it called? Feature? Feature. Expected keyword argument feature. Oh, that's bizarre.
It probably shouldn't be a plot raster. It's plot
oh, excuse me. Thank you. I'm in plot raster instead of plot con. Yes, that won't work well. Okay, there we go. So this is the now it's the number of connections between any two populations. So you can see I2 to E2 has a high number, and these have higher numbers. So yeah, you can also do that for the delay. You know what I mean? If you want to see populations that are further apart with distance based, you know, they'll have a longer delay. What else did we want to talk about? Include the features. Oh yes. You can also group it by population or by cell. Okay, so right now it's grouped by population. Let's group it by cell instead. Group by equals cell. And now I happen to know that this will crash unless we put in feature weight, because the default feature is strength, and we'll have to fix that. So if you use group by cell, the default is not strength, the weight, but it's a bug to be fixed. I didn't do do that. Okay, so yeah, so we're looking at the weight between individual cells now. Yeah, and the default is to order them by GID. So you know when you create a population, you create all those cells at once. So those GIDs are contiguous, right? So the population, this is like grouping by population, or ordering by population. But we can also order by Y, by Z, and those give us different views into the data. So I'm going to copy this, add a new code cell, and paste it. I'm going to say, what is it, sort by, order by. Order by equals Y. Let's do. And not a lot of difference in this particular case. But in many cases, like when in the spinal cord, which we'll continue to build up later, we definitely want it ordered by Y, because we're going to be looking for waves traveling through, right? So you need it ordered by spatially. Okay, order by, group by.
Is there a way to change the axis labels so you can see so you can display visually what population?
Yeah, let's see. You'd have to do it, I guess, manually after the fact. Currently, we're completely revamping our plotting and analysis to sort of separate them and make it real clean and awesome. And once that's done, you'll be able to modify anything as an input to the command line. Right now, it's sort of mostly set, but this creates a matplotlib figure, which you can then go in and change the font size and change the whatever. Yeah, because when you do it by cell, obviously you get a lot of stuff going on there. Okay, so anything else interesting in the raster, the columns? yeah, no, I think that's about it for the plot con. So now let's move on to the rate spectrogram. I got a semicolon, shift enter. All right, yeah, so this is the spectrogram, firing rate spectrogram. So on the x-axis is time, and on the y-axis is the frequency, and here's the power at each frequency. Now, this is obviously very crowded, so let's right away change what we include. Actually, let's point out one thing here. If you put a question mark at the end of this in Colab or in IPython, it'll spit up the doc string. So this is the documentation that's in the code. It's used to build the website, but it's also, it lives right in the code. So in this case, we've got include, but we want to just include all cells, so we just see one, so we can see it really up close. And a semicolon. Where's the hell? Oh, does it need to be a list? I think it needs to be a list. There we go. So now we're just looking at all the cells analyzed at once. And we can see in this simulation that there's a lot of power around 25 or 30 hertz, like we saw when you guys went through this tutorial earlier. And let's, this is what rate spectrogram. I don't know which one that's in, actually. Spikes. Spectrogram, okay. We've got the include. Oh, yeah, you can do a time range. So on most of these plots, on all the plots that include time, you can also set a time range to zoom in on different areas. So let's look at that real quick. New code block. Let's do time. Oh yeah, so it even nicely shows your options here. Time range equals, and let's just sort of zoom in on the first 400 milliseconds there and see what's going on. 0 to 400. Shift enter. All right, now we're zoomed in on 0 to 400. Yeah, that's about it for this. Really, you wanted the time range and whatever you want to include. Plotting the spike histogram. So let's shift enter on this one. This is still in spikes, spike histogram. Yes, so this is, histogram is, the number of spikes in each time bin. Give me 1 sec to see what that's about. Yeah. Okay, back to you guys. Yeah, so this is kind of hard to read. I think the default bin size is pretty low. Yeah, the default bin size is 5 milliseconds. So I'm going to go ahead and mute Slack. If anybody, if a question or something arises in Slack, please raise it to me verbally. Pause notification for an hour. Okay. So let's explore changing the bin size on this spike histogram here. Let's go bin size equals, let's double it and see what it looks like. Okay, a little cleaner now, a little bit different. So let's actually change that to 20 just to explore. Okay. And again, with include, you can kick out or leave in any one of the populations, as we talked about in the other one. The spike hist, time range, again, you can set your time window. Yeah, right now the measure is what is the other measure? Rate or count. The default is rate. Oh, so this is actually, oh yeah, this is the average cell firing rate. Excuse me. This is not a spike histogram, really. We can, let's look at what an actual spike histogram looks like real quick by changing the measure to count. Measure equals yes. Okay, let's new code cell, paste that. Measure is now count, close parenthesis and the important semicolon. Okay, so now this is an actual spike histogram with the spike count in the different bins. Again, you can modify the pop colors, the figure size, save the data, all that stuff. So yeah, spike histogram. Now spike statistics, which is fun, the box plots. So we'll just do the default right away to see what pops up. And yeah, so by default, this actually plots several spike statistics. So yeah, on the left here, you can see that each one of these is a population. And this top graph is the spiking rate, firing rate. I believe the mean is the star, the median is the middle line, and the bars extend to the first and third quartile or something, and anything plus sign is an outlier. So yeah, it gives you a quick way to see the firing rates in all the cells. This next one is the irregularity. So there's this ISI you can use. It's a way to measure irregularity. So if it's spiking real regularly, you're going to have a low value in this. But if it's really irregular, you're going to have a higher value. Great. So yeah, let's explore this guy.
Joe?
Yeah.
Short question. Is possible to upload also experimental data and plot simultaneously the results from the simulation and the experimental data, just to see whether, like, you can reproduce the data? I mean, this function was available, at least was a way to do that in Neuron, and I was wondering whether this has been implemented also in NetPyNE.
So as I talked about, I'm currently, one of my biggest jobs right now is revamping the plotting and analysis to separate the plotting and the analysis. So like a lot of projects that evolve, this tool evolved as something we use in the lab all the time. And so it wasn't really pre-planned. And so the analysis and the plotting are really sort of mixed together, and it's not what we would like it to be. So in the reboot, which should be ready in a couple months, plotting and analysis will be completely separated. And so it will be very easy to, as long as you get the data into this format, it'll be very easy to use these plots with any data, not just directly from NetPyNE. Right now, you would have to get that matplotlib figure, get the data you've got, get it into this form, and then plot it on top of it yourself. But it's possible.
Okay, perfect. Thanks.
Yep. Joe, can I just, I know maybe that you can shelf this question for another time, because I know it's not what this is actually about, but I also know someone told us about how you can just get the raw data, but I've completely forgotten how you can get the data for these plots exported to pandas. Or
To get the data for these plots?
Yeah.
Yeah, that's very easy. You just run the plot. So I'm going to say foo equals SA.plot spike stats. Let's look at foo. It's a tuple. Okay, so yeah, what it returns is foo0 is the figure, the matplotlib figure. So if you want to manipulate it later, you would say, yeah, when you did this, what you can do is, still there? Yeah, you can do fig, comma, data equals the plot. And now we've got, yeah, type fig. It's a matplotlib figure, so you can, edit the axes, add more plots, do whatever you want to it. And then the data, I'll just print it, it might be pretty big, is, yeah, all the numbers you need to make this plot. This is the statistics data. Yeah, I'm not exactly sure what it is, but yeah, the data for every plot should be accessible from the plot. You should be able to get that out. Okay. And yeah, plot spike stats. Clear that output. Okay, and we want to do spike stats now. Okay, so it's got the include again, but this include is a little bit different because we want to have all cells lumped together and analyze the statistics, and we also want to have each pop separately. So the default in this is each pop, all cells, and that if we didn't have all cells, we wouldn't get the average across all of them up here. And if we didn't have each pop, we could do a list of populations, as we talked about it with the earlier stuff. Yeah, time range. Again, you can specify the time range. Yeah, okay, so the stats. So the default statistics that you're going to get out is two figures. One is the firing rate, and one is the irregularity index. But there's also two other measures we can look at, sync and pair sync. So I'm going to copy those and run them. Is there anything else we really want to look at besides pop colors, fig size, all that stuff? No, it's just, yeah, plot spike stats. V, and this is stats. So I want my stats to equal this list instead. Shift enter. Oh, well, let's see if it's one or both of those that don't work. Okay. So, oh, yeah. It's not installed by default, but oh yeah, we should be able to do it. Well, we'll just try it real quick here because you guys might want to do this. I believe I can just pip install PySpike. Is that true? Oh yeah. All right. So we'll pip install PySpike. So exclamation point pip install. Shift enter. Error failed building will. Accept error. Oh, okay. Maybe this is why we don't include it automatically. It seems it's not functional. PySpike is not installable right now. Yeah. So yeah. But anyways, this PySpike allows you to calculate synchrony measures. between groups of cells. And yeah, maybe we'll want to implement it ourselves and not rely on PySpike. Anyway, so yeah, so those are all this, you get a bunch of interesting statistics out for these cells and plotted nicely. Yes, so now plot traces. So this one is, probably the one we use a lot. It's a very common. You want to see what your traces are doing. Here we go. Okay, so I just did plot traces with no defaults, with no entries. And so in that case, it's basically going to print every trace that it has recorded. So if we look at our GUI 2.3, where we record traces at the bottom here. Okay, so the traces we were recorded, the voltage of the soma, potassium current, and then 2 calcium concentrations. And we did that for, I did that for the first cell of each population. To analysis, traces, analysis, traces, traces. Okay, and yeah, so by default, it's going to print everything it has. So this is cell 0, so it's plotting it by cell, not by trace. We could also have all the VSOMAs lined up next to each other, right? So this is all the stuff that's going on in cell 0, cell 10, the first cell of population I2. Great, so it's all the plots for all those cells grouped by cell. The other way you might want to see these, though, is grouped by trace. If you want to compare the traces directly, then you want to by trace instead of by cell. Oh, 1 fig per, it's called, sorry. 1 fig per. The default is 1 fig per cell, but we're going to now try it 1 fig per trace. Okay, let's try that. Great. So now we see the first plot is all the VSOMAs. The next plot is all the CAI, all the CAO, and all the potassium currents. Okay, so you can plot it by cell, 1 fig per cell, or 1 fig per trace. Now, so these, the default is going to be not to overlay them, but often it's nice to overlay. Oh, it says the default is true. That doesn't look overlaid. I might have to update this. But yeah, so let's set overlay equal to true. Copy that and keep it the traces. Overlay equals true. Oh, there we go. Nice. Yeah, so now you can see we did it by trace, or one fig per trace. That way the traces are all the same when you overlay them. That's what the soma looks like. Here's the calcium. Yes. So traces per cell, trace, time range again. Overlay colors, you can switch. You can turn the axis on or off, which is kind of nice for a lot of these figures. And then it'll add a scale bar. So yeah, let's do that real quick. So it's false, shift enter. Yeah, so now you can see we don't have that axis anymore, and it adds a scale bar to the figures. And yeah, I think that's all we want to talk about with the traces.
is it just randomly selecting one cell per population there?
No, we specified 1 cell per population in our sim config.
Okay. Sorry. Yeah. So everyone you recorded.
Yeah, exactly.
Okay.
So yeah, by default, it we can whittle down, just plot this cell, just plot this trace, but by default, it's going to do it all. And yeah, we set it up with this is a nice way to get the first cell from each population, a list of tuples. And so yeah, it can only plot what we recorded. So that is what we recorded. Okay.
Can you also take us through plot raster? Because I think one of those entries were deleted while you were...
Oh, thank you. Yes, plot raster is a very important one. Let's do that one now. Thank you. So we'll do sa.plot raster, semicolon. Great. So yeah, this is really a workhorse. Probably the plot we use the most is the raster plot. So that's going to be in spikes, and that's going to be plot raster. Again, include, we can leave populations out if we want to. By default, it plots them all. You could even do single cells and stuff if you wanted to. Really, with a raster, it's kind of you want to see everything. We can adjust the time range. Yeah, the maximum spikes comes into play when you're doing really large simulations. If there's like 1 billion spikes and you try to plot it, it's not going to work very well. So if you set a max, it has a default actually. Order inverse. Okay, order by and order inverse are important. Pop rates. Oh yeah, we should turn on pop rates. We could add a spike hist. We could do sync lines. Yeah, there's some fun stuff here. Okay, so first let's do order by. So by default, they're ordered by GID. But you can also order it by Y or by any spatial variable. SA.plot raster ordered by equals. So yeah, now you can see the cells are interspersed because they're ordered by their vertical position. So order by. Okay, order inverse comes into play a lot, like here. So for example, you can see that E2 is first in the legend, but it's at the bottom of the file here. It's because the y-axis typically goes upwards in value, but we start the GIDs. We start at the top of our models usually and go down. So order inverse allows you to correct for that. So I'm going to copy that and put in a new cell here. Yeah, what was I just talking about? Okay, yeah, I want to keep that the same now. Oh, order inverse, excuse me. Order inverse is true. Okay, and now we can see that E lines up, I is below it, E4 is below that. It makes the plot a little nicer. Well, and it, more meaningful because we're defining cortex going downward, and so it's physically laid out the same way as in reality, which is always helpful. Okay, so order by, order inverse. Labels, yeah, we can do a legend or an overlay. Let's do the pop rates here. It's true, and this will add a little more information to our figure. Now we can see the average firing rate of each population here in the legend. And the overall firing rate is always above the figure here. Yeah, instead of a legend, you can also do an overlay, which is nicer in some instances. Where is that? Is that a legend? Labels. Yeah, labels we want to be overlay instead. Okay, so I'm going to do this. Set labels equal to overlay. There we go. So now they line up next to the population of interest in their nice color. I think we can turn the axis off on this one too. No, maybe not. Okay. So yeah, that's the overlay, the pop rates. Okay, we can put a spike histogram directly on top of this, and we can also plot those sync lines. So let's do the spike histogram. Do it over. Back here. Yeah, and we want spike hist to overlay. The other option is subplot, which would make it a separate plot underneath it. But if we overlay the spike histogram, now you can see, yeah, that's the spike histogram right on top of the raster plot. So you can, it's kind of fun. We can try that as a subplot. So, and now it's below. Oh, that's weird. This is a bug. Okay, it inverted that axis and not this one. So that is something we'll have to fix. Oh yeah, so sync lines is the one other thing that's really of interest in here. Sync lines. Yeah, sync lines. And again, this is no real analysis or anything. This is a purely visual way of sort of seeing the synchrony. So if you've got dark reds with clean whites in between, you've got a lot of you're seeing a lot of synchrony. If you don't, you're going to have sort of an overall pink all the way across. We can see some pretty decent synchrony in here. Makes it easier to see when you put a red line on every spike. Okay, I think, marker. Yeah, marker's fun, and marker size. Yeah, let's do that. Okay, let's copy this, add a new block. Look at the sync lines. And you can use any of the matplotlib markers, so like 0, asterisk, all sorts of that stuff. Yeah, so now it's circles instead of lines. Using the marker size option, you can make those bigger or smaller depending on how big your simulation is and stuff. You'll probably need to adjust markers and marker sizes. Yeah. Pop colors. Yeah, let's just play with pop colors once real quick. We haven't done it on any figure, but let's just let's do that now. So we can set our own population colors, because we got half an hour left and only 2 more figures. Great. So yeah, so let's set up our colors. And like it says, pop colors has to be a dictionary with a custom color value used for each population key. Okay, so colors is a dictionary. And we've got E2, I2, E4, I4, E5, I5. So E2, oops. E2 is going to be red. How does this work? Colors is a dictionary. Let me try this. Okay. And then colors E4. I'm not even I'm not doing that right. Equals pink colors E5 equals and I think it'll work with these colors, but I'm not totally sure. We might have to look up hex values. Oops. Colors. To something like blues and purples. Four. Five. Okay, so now we've got a colors dictionary. We should be able to feed that in and affect the colors there. So let's just copy this. And then we want to call that pop colors. Aha, cool, it worked. So you can set custom colors for all your populations. And yeah, so it's actually a nice thing to do if you're doing a bunch of stuff, is to set up that for your project, and then every analysis function here will accept that pop colors dictionary, and you'll get all your population colors the same across all of your analysis. Great. So that's it for plot raster. Any other questions on there? Then we'll move on to plot LFP, local field potentials. Oh, that seems to be successful. Okay, and yeah, by default, this is going to spit out several figures. There's an exception in plot shape. Oh, plot shape is just one of the plots, the bottom one. Oh, and it actually made the plot, so I don't know what the crash is, but we'll have to look into it later. Yeah, so you can see it generates a bunch of figures by default. We have, let's look at our LFP setup real quick here. So LFP, set up LFP. Here we go. Simconfig dot record LFP is what is that doing? Okay, it's -15y 1 times z. That. Well, anyways, we end up with 3 LFP electrodes positioned using this this method. And yeah, the default plot will create an average of those and show you them individually. A nice scale bar there. The spectral density average over all of the electrodes, and then for each electrode individually. And that's how much power is in each frequency. So in population, it's got a lot of power at that 2530, but also a lot of high frequency power, whereas the other 2 populations don't have much high frequency power. Yeah, this is the, what do you call this, the spectrogram. So again, it's frequency on here. This is time, so it's how the frequency is changing over time. And yeah, this is plot shape. This uses neurons built-in plot shape. It seems like there was some sort of problem. But yeah, let's see if we can figure out what all these figures are. So we're going to go to analysis.lfp. Plot LFP is the only one we got in LFP. We can set a time range. The electrodes default is average and all, but you can I believe you can also select. I don't know why you would create them in the config and then not use them, but I believe you can. Yeah, the default is to use all of them and to take the average. If you don't want to take the average, you would just say electrodes equals all in a list. Yes, so here's the plots. So by default, it's going to create 4 plots: time series, power spectrum density, spectrogram, and the locations. And yeah, there's lots of options regarding analysis of the LFP. So you can smooth, whether to include the axon or not. A lot of times, these are good to have on log scales because it varies so widely in scale. So you can set the log scales. You can normalize stuff. You can set a filter frequency. Yeah, so I don't actually know much about LFP, but yeah, they got all these different options you can do because you want to tweak the analysis a little bit. Is there anything you want to play around with here? No, I think this is pretty much it. We could, adjust the time series, the time range and stuff. But yeah, it's about it for LFP.
What's the last plot showing? The location.
Good question. I'm guessing that color is related to the voltage internal. But I would actually have to look at the code to see because it's not entirely clear here. But yeah, let's take a look. So plot LFP. It's hard to read. P. Okay, so plot LFP. Okay, yeah, that's the time series plot. There's the PSD plot. Here's the spectrogram plot. And locations plot. Here we go. For each cell, recording electrodes get transfer resistance. Okay. That apparently is the transfer resistance to the electrodes. Again, I don't know much about LFP, so I'm not sure how to translate that for you. Yeah, anybody work with LFP? Do you have any idea what transfer resistance is about?
Given that it's called locations, maybe it's correlated with how near the probes are to the cells.
Oh, yeah, it must be. Yeah, you're right. Yeah, I'm guessing this plot is supposed to have axes labels, which didn't show up because of the crash. But yeah, I'm guessing, yeah, the electrodes are going to be over here. Yeah, I'll bet it's based on the distance from the electrode. All right, so yeah, that's local field potentials. And yeah, the last thing we're going to look at is plotting the reaction diffusion concentration. This will eventually have it so it'll just find the first species label and the first region label and plot it so you don't have to put anything in. But if you don't put anything in right now, this will crash because it requires these inputs. Let's take a quick look at the RxD stuff in here. Great. Okay, so we got our RxD params, initial concentrations, diffusion coefficients, all this stuff. We got 3 regions, cytosol ER and site ER membrane. And we've got, looks like 2 species, calcium and IP3. So yeah, let's just plot. This is going to be calcium in the ECS. Regions here, ECS, extracellular. also got ER, cytosol, cytosol ER membrane. Great. So let's see. It looks like we've got 1, 2, 3, 4 regions and 2 species. So let's go ahead and plot every combination of those. Do something to make you think on your feet here. So let's we'll do a for loop. So let's do 4 species in one of our species, CA P3. And then for region in the first region was cytosol, and then we had ER, and then site ER underscore brain. We've got the ECS, the extracellular space. Oops. Then we want to, SA.plot. I'm just going to copy this down to here, paste it. So plot RxD concentration, the species label equals species, and the region label equals region. And let's see how that goes. No such region. Well, yeah, and again, I'm not, I don't do much with RxD, and I don't know much about the theory and everything. But yeah, you can get out these reaction diffusion concentration plots. Yeah, let's see, it's 4:40, so we got about 10 minutes left here. Any figures or analyses that anybody wanted more detail on or to talk about or anything? No? All right.
Yeah. I asked this before. So let's say if you've recorded data from in NetPyNE, so you've set up you've recorded your LFP traces, for example.
Yeah.
What's the correct way to access the raw data? Can you do it without plotting it, or do you have to plot it and then get the data?
No, you can get the raw data from SIM. So sim data. Oops. Yeah, sim data.
That's what I couldn't remember.
And yeah, so in our sim data, it's a dictionary. SPKT is just a list of the times for every spike in it, and then the spike ID is the GID that aligns with that spike time. And then, yeah, then every trace that you recorded is going to have a dictionary. So VSOMA is a dictionary that has each cell that it recorded VSOMA for inside it. And yeah, so all the data that gets recorded, you can find after the fact in sim.simdata, and then you can do your own plots and your own different analyses and all that. All right, well, let's take a 20-minute break then and come back at 5. All right. I'll see you guys at 5 o'clock.
Thanks.
Yeah, thank you.
Thank you.
Perfect. Thanks.
Great. Stop recording.
Thank you.
Yeah, thank you guys.
Thank you.
Stop recording. The recording will be saved in Solidar Durability Drive. Oh, great.